as a scientist, we want to have the ability to isolate one specific thing so that we can focus our study on that one specific thing. With bacteria and the fact that they are so small, you have to have a way to purely isolate one specific type of bacteria. So, the purpose of the pure culture technique is to separate one pure sample from a mixture. You can see here that I've got kind of cream bacteria and yellow bacteria and orange bacteria. Obviously, this is not a pure culture. So when it comes to being able to um, diagnose specific characteristics to one type of bacteria, let's say the gram stain or the motility test or even the shape of the bacteria, you have to have pure cultures. If you have a mixture of, let's say, bacilli and cocci, you've got the rod-shaped bacteria and you've got the circular cocci bacteria. So how do we do this? Why do we do this? Well, primarily because if we use a mixed culture, we get mixed results. And you don't want that. You want, this is the bacteria, this is what the result is always. If I do it 78 different times, because this is the only bacteria in my sample, this is the result I get. It would literally be impossible to check which bacteria you're working with any accuracy unless you have a pure culture. So if you had something like this, and let's say I took from here, see how they're all three touching the yellow, the kind of beigey, and the orange? There's nothing to say that they're not mixed up in that particular colony. How do we do this? You're going to obtain a mixed culture of bacteria, basically in liquid media. You're going to obtain a nutrient agar plate. They've already been made. All you need to do is get them. And you're going to use a Sharpie on the bottom to basically put at 12, 3, 6 o'clock, and 9 o'clock markers. So you can do 1, 2, 3, 4. You can do 1, 2, 3, 4. But you need to have markers under the plate so that when you're looking at it from the top, you can see those markers underneath. I always think of north, east, south, and west, and make sure you label the plate as yours. Okay, and this is all with the plate inverted. In other words, the agar is actually sitting over here. I want a pure culture. I've got to sterilize my inoculating loop. So you flame and cool your inoculating loop. With the loop, obtain a sample of the liquid culture and dot it in position 1. Now, I'm trying to kind of spread these bacteria thinner and thinner every time I do a streak on this plate. So if I have an inoculating loop that still has bacteria on it from when I first harvested, I'm going to have a lot of bacteria. And I don't want that. I want it to basically be fewer and fewer and fewer every time I go from 1 to 2, 2 to 3, 3 to 4. So in between these steps, I will always flame and cool the loop. The mixed culture, I'm done with at this point. I literally just put one dot of that culture at position 1, and that's it. Using my sterile loop, you are going to streak, you're going to basically on your plate take your loop and basically go from the dot of bacteria over to the next number. So that was number 1. Here's number 2. I've flamed and cooled my loop. I'm going to go and drag this toward number 2. And I'm going to do it 4 or 5 times. Just kind of depends on what you want to do and what works best as far as the technique goes. Now remember, I don't want any extra bacteria from what I've just placed in position 2. I'm going to flame and cool my loop again. I've just flamed and cooled my loop. I'm going to streak again, but instead of starting at position 1, I'm going to start at position 2. So at position 2, I'm going to take my newly sterilized, cooled inoculating loop and drag from number 2 to number 3 4 or 5 times. Flame and cool your loop, because any remaining bacteria that are on here, I want them dead. I'm trying to thin out the herd here. I want to make sure that I'm taking any extra out because I want as few as possible to end up in this last streak that I have here from number 3 to number 4. So flame and cool the loop. Using the sterile loop, you're now going to streak from position 3 to 4. Now remember, every number from 1 to 2, 2 to 3, even 3 to 4, I should have fewer and fewer bacteria that I'm picking up and pushing along this fresh, clean nutrient auger. By the time I get to 3, I should have maybe 4 or 5 that I'm streaking. Why? This is why. Once you've done the third streak, basically keeping the plate as inverted as possible, you wrap the edge of the plate in parafilm. So parafilm is like saran wrap, only it forms a tight seal that actually prevents airflow inside of my plate. Now, you always want to keep it inverted. Why? Because moisture eventually will get into there. And if you've got it right side up, just like on a boiling pot, all the moisture gets on the lid. The last thing you want is for that to go back onto the auger and spread everything out. So you keep it inverted so that if there is any moisture, it stays at the bottom and can't actually get on the plate that you just made. As far as what you do next, normally you're going to place this in an incubator for 24 to 48 hours. The next day or 2 days later, depending on how your lab works, you're going to look at the plate and see if there's any growth, and specifically see if you have isolated colonies. So if you look at this one probably had five, no, actually it still had 4. My bad. This is where the bacteria is thickest. That means they probably put their droplet of bacteria from their original culture up here. And then they streaked with their newly sterilized loop until they got to position 2. Flame and cool your loop, streak. So that's 2 to 3. Okay? Notice that this and this almost look identical, right? Even though technically I am lowering the amount of bacteria in this area versus this area. You'll notice that there are some really, really thick bands here that aren't necessarily here. But just naked eye looking at it, this actually does look very similar. But as I've pushed this in this direction, it's thinned out the herd. Flame and cool your loop again. Now you're going to go, there's position 1, 2, 3. You're going to go from position 3 to position 4. You'll notice now it's become really obvious that there are a lot fewer bacteria here than there were here. I was right. It is 5 streets. Sorry. So 3 to 4. Now in position 4, you're going to, again, flame and cool your inoculating loop. And then you're going to drag your bacteria this direction. Now, each of these isolated colonies, we talked about this in lecture, and we said that when you've got one colony, it's the start of a pile of bacteria that literally started from one individual bacteria. So what do I have here? In each of these colonies that is all by itself, see this guy that's kind of doubled up? I don't want that one. I want somebody who's like literally hanging out all by himself, nobody near him, nobody touching him. That's how I guarantee that the bacteria I'm looking at is a pure culture. Because if nobody's around it, that all started from one sample of bacteria. And that is the pure culture technique.